Hi, Gen Bio. This is Dr. Fry. I'm here with our week 2 lab. And again, just like last week, we're going to split it into 2 parts. So today's part will be on, well, the first part will be on scientific investigation, and the second part will be on dilutions. Okay, so the first video will just cover scientific investigation. And we are going to look at that in lab this week using Sowbugs and Pillbugs. So you're probably familiar with Pillbugs. These little critters can be found outside in the garden, in the backyard, and they curl into a little C-shaped pill, or even almost like a ball, like into a pill shape, when they are nervous or hiding or just trying to shelter themselves. Sowbugs are closely related, and we're going to look at the environmental preferences of these little critters in lab today. So before we look too much at the Sowbugs and the Pillbugs, I want to give you an introduction on the scientific method, just to make sure we're all on the same page with terms and knowing how to describe our process and how to go about setting up and designing a good experiment. So most of research begins with making observations. You can make observations about the world around you all the time. But from that point, you need to get to identifying questions. So posing a question that's clear and definable. So just for example, I'll give you an example scenario to work through this today. A question that you might have is whether or not air pollution affects the production of plants. You could pick a specific plant, such as a soybean. Does air pollution affect soybean production? So it's a very clear question. It's definable. We know concrete, we're talking about a specific plant, and we know what we want to find out about that plant based on its exposure to air pollution. And then we need to generate a hypothesis. So a hypothesis will propose an answer to your question, and it needs to be testable as well as falsifiable. So we typically say that you can prove your hypothesis false. You might not be able to prove it true because there could be extenuating circumstances that are not able to be realized by the researcher. We cannot put our finger on every single condition that might affect the result. And so we can say we could prove a hypothesis false, but we can't always prove it true. And then from that point, we need to suggest a null hypothesis, which means that there is no difference between the test groups, and an alternate hypothesis, which means that there is a difference between the test groups. I'm talking. And then, so the null hypothesis would be that there would be no effect, for example, there would be no effect of air pollution on the production of soybean plants in our example. The alternative hypothesis would say that yes, there is an effect of air pollution on the production of soybean plants. Okay, so this would be suggesting that the alternative hypothesis would be suggesting that there is a difference caused by air pollution, or whatever the example that you are studying is. And then you need to make a prediction. So if you think that the hypothesis is true, then you predict the results of your experiment. So the results would indicate one way or the other whether or not your hypothesis is correct. Okay, so from that point, we need to start designing our experiment. We need to determine we need to pin down a few variables, okay? So the variables that you need to know are the independent variable, the dependent variable, controlled variables are the top 3 key ones. So an independent variable is the variable that you, as the investigator, as the researcher, are manipulating, what you are changing to observe a result in your experiment. So in this example that we're looking at, the independent variable would be exposing the soybean plants to either filtered air, which would be baseline, the default, or say sulfur dioxide, which would be your air pollutant. Okay. And then the dependent variable is the variable that you will measure after exposing these soybean plants to either the filtered air or the sulfur dioxide. So what you measure, what you're going to count, what you're going to analyze, that's your dependent variable, okay? So in this example, it could be any number of things, such as the number of bean pods produced by the soybean plant, the number of seeds per pod, or the pod weight, for example. So any of these things could be dependent variables that you as the researcher would then examine to determine the results of your experiment, okay? And then you will have controlled conditions in every experiment. You need to hold everything else in the environment of these soybean plants constant so that you really focus on the one thing that you change, the independent variable. The one thing that you are manipulating needs to be isolated. And so everything else about these plants has to be held the same in each of the groups. So for instance, in this example, we would have the same exposure to light or dark for each group of soybeans, right? So that would be a controlled variable. You would also want to give them equal amounts of water, equal amounts of nutrient, same soil content. So all of those things would be things that you should look at and keep constant in between the two groups. So that would be your controlled variables, okay? And then other terms that you should know to design your experiment are level of treatment. So the level of treatment is applied to your independent variable. And this would be something that you would research ahead of time before making decisions. It's the amount of whatever your independent variable is. So it could be the amount of sulfur dioxide exposed to the plants and for how long. And this is something, like I said, you would look at previous papers to determine what had been done, what hadn't been done, what's like an optimal range. If you were looking at enzyme activity, you would want to look at previous research to determine a starting point for that, for the level of treatment. And then you always need to have replications. So each experiment needs to be done more than once. So we often in the lab, we would usually do things in triplicate just to start with, because we want to make sure that it's not just one fluke that we're observing and then making a conclusion from. You need to make sure that your results are reliable by replicating it a number of times, okay? And also, along the same lines, you want your sample size to be more than one. Sample size is usually indicated by a small lowercase letter n, okay? And so the sample size of 1 really doesn't mean anything, because it could be just one circumstance that you see a result, and then in the next circumstance you see a completely different result. And so that is not a reliable test. You want to have a larger sample size, the largest that you can have really within the confines of practicality is best. Okay, and then one other term that you should know is the control. The control condition is different from controlled variables. You want to have a control condition, which is an established normal result. So you would have a positive control, which would show the result that you would expect. So if you know that soybeans will produce poorly if given some other air pollutant because of previous research, you might want to include that to make sure that your soybeans are behaving just like all the other soybeans should. And that would be a positive control. It gives the result that you expect to see. And then a negative control would be the opposite. It establishes that there's no, again, no change in these soybean plants that for one reason or another would make them unhealthy and not able to produce even in normal conditions, in healthy conditions for a soybean plant. Okay. All right, and then after you've gone ahead, designed and completed your experiment, you need to analyze the results. You need to make this data into tables, graphs, write it up into a paper. Make sure you do statistical tests to prove that there is, in fact, some conclusion that can be gained from the results of your experiment. So we're going to be doing a lot of that in the lab this semester, making tables. And then next semester, we have you write up, just like you were writing a scientific journal article, just to get these results that you discover out into the scientific community, to share that information with the community. And as I mentioned, we'll get more into this towards the end of the lecture here, you need to do tests to statistically support your conclusions. You can't just claim, well, I saw that there seemed to be a trend, and I think that that means that X, Y, or Z. You need to have statistics to back it up. You need to say, look, I quantified the results and there's something here, okay? All right, and after that, after you make your results presentable, then you can make conclusions. Now you wanna be a little bit careful with making conclusions and interpretations because, as I mentioned, there are things that we as researchers can't always consider everything, right? We are not aware sometimes of a factor that might be influencing our experiment. So you want to make a conclusion, but you don't want to claim that you know everything, you know. So make an interpretation, show your results, statistically support them, and present them to the scientific community, either through a conference or a journal article, a meeting. You want to share those results with the scientific community, okay? All right, so let's move to the next slide here and take a look at Sowbugs and Pillbugs. Like I said, these little critters can be found outside in the garden. They're little small bugs that, as you can see in the bottom picture here, the Pillbugs can curl up into a little pill. Sowbugs can't really curl up as much as a Pillbug, but they can form like a small C shape. But they're closely related, and for the purposes of today's labs, we're going to use them interchangeably. So they're both in the class Crustacea and the order Isopoda. They get to be about a little over a centimeter, and they like to live in damp, dark areas where they have access to decaying plant material, which is their food, okay? And they each have 7 pairs of legs, overlapping plates, as you can see on the pictures here. You can tell the difference because Sowbugs have 2 tail appendages, and the Pillbugs do not. And then they can't curl up as much as the Pillbug. So the top picture here is the Sowbug, and then the bottom picture is the Pillbug. Okay, but again, we're using them interchangeably. And we're going to look at the environmental factors that are the preferred habitats of Sowbugs and Pillbugs. So environmental factors can be a wide range of things, and they can be either abiotic or biotic. And of course, biotic, as you might take from the name, is anything related to other organisms or life in the environment, okay, bio, right? And then abiotic is something that's not directly connected to a living organism, such as temperature, moisture, light, dark, etc. Okay. And then these factors, these environmental factors, whether abiotic or biotic, can occur in a gradient. So it's not often like you'll see hot in one area, very hot temperatures, and then right next to it cold. You're gonna have a gradient, right? It's gonna be gradually changing from a hotter climate to a cooler climate. And so this is an example of a simple gradient. One factor, my example is temperature, that changes gradually over space. Complex gradients then involve more than one factor. So for example, elevation is one factor that actually involves multiple issues. Elevation changes, so you might be going from like a valley to a mountain, are going to involve changes in temperature, humidity, probably precipitation, soil depth, soil quality, wind, water, lots of things. So all of that is combined in the concept of elevation changes, and that would be an example of a complex gradient where more than one factor changes with elevation. Okay, so for today's or this week's lab, you're going to be looking at changing factors about the Sowbugs and the Pillbugs environments to see if they have a preference or a desire to migrate to or live in the environment that you create for them. Okay. And if you were doing this as a long-term study, you would need to then measure over time what indicates the Sowbugs and Pillbugs' desire to live there. We're obviously going to keep it relatively short for the lab this week, but for example, you could look at how much they, if you were doing it long term, you could look at how much they grow, how much migration takes place, how much reproduction takes place in each of these environments, because obviously if it's a more fitting environment for whatever organism you're looking at, they're probably going to grow, they're going to migrate more to that area, they're going to reproduce more, and their offspring will be more viable, and that would be overall a better living environment for them than a less ideal environment where the environment, the characteristics about the environment are not as beneficial to the growth or the reproduction and viability of these animals. Okay. All right, so let's move to our next slide then. This graph is taken directly from your lab manual. On the x-axis, you'll see that there's symbols representing different qualities of the environment, different environmental variables. So you might have a range where X1 here represents a hot environment, warm would be in the middle, and then a cold environment would be on the other end of that x-axis. On the y-axis then is the response of the organism to that environmental factor. So either the presence, like the organism has migrated more to that area, growth, reproduction, all the factors that we just mentioned. And basically what you will see then is that the peak of the curve is indicating the greatest positive response of the population. So whatever it be, if you're measuring their migration, their growth, their reproduction, whatever the variable is, you're having the greatest positive response in the preferred environmental characteristics. So in this example, a warm climate is the most preferred environment for whatever the response is, so growth, reproduction, viability, etc. Okay. All right, so let's move on then to our next slide, scientific process. We need to always come back then. Today, we're looking at our question is going to be, what kind of environment is the preferred habitat of Sowbugs? So you want to make a specific question. You want to start with one specific variable just to keep it simple. We're going to do simple gradient first, okay? Do Sowbugs prefer light? Do they prefer shade? Do they prefer pitch black? That could be one characteristic, the amount of light that you could alter to determine which condition is the preferred habitat for your bugs today. And then that's just an example. You will pick your own characteristic to examine based on what we have in lab. So in lab, we have other things. We can do temperature, we have ice and hot plates. We can do different amounts of food or different types of food that we will have available for you guys. And there's a slew of other options that you can choose from: moisture, soil depth, access to water, etc. So you will think about and design your own experiment, and then you're going to expose your group of Sowbugs to that environment for a period of time, and then see what happens, see where they migrate to, which seems to be the preferred environment for them, okay? And then after the experiment is over, we are going to use statistical testing to analyze the results that you observe. So I said you always need to have some numbers, some quantifiable data to back up your claim. And so the test that we are going to use is the chi-squared test. Chi-squared test is going to compare your observed result with an expected result. So for the null hypothesis, the expected result is that there will be no preference for any of the conditions that you create. So say you have 1/3 of your environment is light, 1/3 of your environment is dark, and 1/3 of it is shaded. If there is no preference, there should be equal numbers of bugs in each of those environments. And that would be our expected result. We have to expect the null, that there is no preference, okay? So for the chi-squared test, we assume that there's no preference. There's going to be no change at the end of the test. And basically, if you see a change from the null, then we have some numbers to back that up, because we're expecting baseline. We're expecting everything to be equal, and that those Sowbugs don't have any real preference between light, dark, and shade. But if you do, in fact, see that there's a greater number of Sowbugs in, say, the dark condition, then you can say, hey, there's something out of the norm here. That my observed result does not line up with the expected baseline null result, okay? So what you then will say is that you will do some calculations to show that there's something out of the norm here, and that I can back that up using the chi-squared test, okay? So all the words on this slide are basically saying what I just told you, is that there's going to be no change or minimal change, not enough, that change couldn't be due to chance. So random chance, well, you might get, if you had 30 bugs total, you might get 11 in one and 9 in the other and 10 over here, but that change isn't large enough to make a conclusion. It could be due to chance, right? So we have to set aside there a little bit of room for error, that, look, some of this change that we see could mean nothing, right? All right, so then that brings us to the word significant difference. In scientific writing and in meetings or poster presentations, if you say there's a significant difference between your results, that means it has been supported by a statistical test. You can't just throw around the word significant like we do in everyday language. Oh, I saw a significant difference. That means something in science writing. It means that you did a statistical analysis and you saw that there was a difference, okay? So then you would say that, look, my results don't appear to be due to chance. And traditionally, statistical significance is defined as 5% or less frequent occurrence due to chance. So say you roll a single die 600 times. There's 6 faces on your die. Is that right? Yeah, 6 faces on your die. And you roll it 600 times. Theoretically, each side of that die should come up 100 times equally, right? But if you see a huge difference from that one side is coming up 95% or more of the time, then you can say, look, there's something else going on here. That die is weighted or something. So there's something out of the norm, and I'm going to conclude that this is not just due to chance, right? So that's what we're basically supporting when we do statistical analysis of our experiments, okay? So the chi-squared test is the test that you'll be using, the statistical test, and it is, the equation for that is chi-squared equals the sum of each of your observed values minus your expected values squared divided by your expected values. So for each of your conditions, say again you have 30 bugs and you have 3 different conditions, each of those you have to take as its own separate calculation. So for instance, each of the expected values here, right here, would be 10. You should expect there, if you had 30 bugs and 3 conditions, there'd be 10 in each, assuming that there's no preference. That's our expected, right? So you should expect there'd be 10 in the dark. And say you actually see your observed value, say that's 15, okay? Then you take 15 that you saw in the dark minus 10, which is our expected, square that, divide it by 10, and then you go and do the exact same thing for the shaded environment. Say you saw 10 in the shade. Now you'd say the observed is 10, the expected is 10, again, square that and divide it by the expected, which is 10. Okay, and then that would leave you with, if I'm doing my math right here, we had 5 left. 5 would be your observed value for the light, minus 10 squared divided by 10 again, and then you have to add all those together. That's what here the sum of each of those means. And so then once you get, at the end of that calculation, once you get your chi-squared value, you have to compare it to a chi-squared distribution table, which is going to be on our next slide, and it's also in your lab manual, and you can find them elsewhere as well. So a chi-squared distribution table, you need to have your chi-squared value, and you also need a degrees of freedom number, which will be the 2 pieces of information that you need then to look at your chart and determine if you have a statistically significant result. So then that will give you a p-value, which is the probability that the result is due to chance. And if it is greater than the critical value for p equals 0.05, which again, we get that number, that p-value, the probability of 0.05, meaning that 5% of the time, we would not see that result, assuming that it's outside of the norm. It's not due to chance, right? So only 5% of the time do we see that result due to chance. Okay, would mean that there's a statistically different result from what the normal range would be. Okay, you would have a wide range of normal, and this is outside of that. Okay, so that's what would happen like 5% of the time if it were due to chance. We as scientists say, okay, that's an acceptable number to say, I'm pretty sure that this result is not due to chance. Okay, so let's go look at our chi-square distribution table here, the next slide. Oh, I skipped over one thing. Let me go back. The degrees of freedom, we use the number of categories minus 1 as the standard number for our degrees of freedom for this test. Okay, so that's something that we need. We have 3 different categories. So we had light, shade, and dark in that example. And so that's 3 categories, minus 1 gives us 2 degrees of freedom. So we then look at our chi-square distribution table here, and you go down, you select 2 degrees of freedom, okay? And you look down at your P, your probability of 0.05, because that's by convention what we agree as scientists is kind of the cutoff. And then that gives us a number of 5.991. And we need to compare our chi-squared value to this number, to 5.991. And if our chi-squared value is larger than 5.991, then we can say that it is outside of the realm of results due to chance. It is statistically significant then. If it is smaller than 5.991, when we do that chi-squared equation, when we do that calculation, then we can't claim that it's due to chance, that it's statistically significant, because it might be due to chance. Okay, so that's how we use a chi-square distribution table, and that's that wraps up our introduction to the scientific process. So the next video that you will watch is on dilution. So that's the second part of our lab for this week. Okay, so I'll see you in the next video, and if you have any questions, you can always email at cfry@lvc.edu. Thanks a lot.